have 2 lectures and 3 practicals. Going to be a long day. Let's get it started. I think all of you are here. Anybody identify your neighbors? Okay, right, all right, yeah. So, the first lecture is by yours, Ramakrishna. Please start your lecture.
Okay. Thank you, Kumar. Good morning to all. Before I start with the lecture, I just want to check in. So the last 2 days, I'm looking at the technical people until they are done. Is it finished? Okay, good. So I'm just checking in with everybody. You had a lot of information. So last night we were also sitting together with the team, and we're just discussing okay, there's quite a lot of information. There's the theoretical background in the lectures, right? And then there is background when it comes to the bioinformatics, right? Which is, I think, for many of you is new. So I'm just checking in terms of the amount of information. How are you doing? Is it too much? Is it too little? Too complex? Too much? Can you give us some feedback? Because that will help us also to determine, how much more information that we're going to present to you in the coming days. It's important for you, especially. Is it okay so far? Yes. Yeah. So think about it also as we are trying to provide you with sort of like the buffet, yeah, of information about microbiomes. And I think, also considering the diversity in research topics that you are working on, I think each of you will probably select, some pieces from the buffet, right, that we provide. The other thing is that sometimes, you hear, at least that's how I always experience these types of courses. There's a lot of information, but what always confuses me, there's a lot of terminology that comes to you. So when you think about the bioinformatics, right, there's a lot of terms that are new to you. So, but I think the most important is, and Victor and Guillermo and Luisa always try, especially with the bioinformatics, to summarize at the end. Okay, there's a lot of information, there's a lot of packages, there's a lot of R scripts, but please keep in mind, like for example, yesterday, that the quality check of your sequences, the filtering, before you go into the analysis, the rarefaction, right, the normalization, that is one of the most important steps before you go into any, data analysis and sort of like interpretation of the data. So try to separate those two things. So there's the details about running these things and looking at it, etc. But try to look at it like quality checks, and then those are important steps. Okay. Good. So the other thing in terms of information, and again, it's all about the buffet and what you would prefer. So many times we say during the lectures, we were also talking about last night, if you want to know more, that's how we say it, please have a look at this article, or have a look at this website, or have a look at this movie, right? Again, it's just providing you with the information, and be selective for yourself, for your PhD. And many times, maybe you're not going to look at a specific element that we presented now, or maybe in the coming year, but store it in your mind, because maybe at some point in your PhD or your master project, you think about, I've heard about that in the lecture, and then I'm going to go back and then look at it again, and then, because when you use it and when you work with it, then it starts to become much more internalized, right? So, Okay, that was, so today we have, like Kumar said, we have the lectures this morning. I think what we did yesterday is to basically set up the computers for this afternoon. I think we will do that again before lunch. That saves quite some time. Then after lunch we will go to the lab, to do the wet lab practical. So we're going to count the colonies, isolate the bugs, and do some assays. And then we come back here, and then we have the computer practicals. And today the topic is all about assembly, right? Microbiome assembly. In terms of assembly, yeah, it's an easy topic, and at the same time it's super complex. And I've been struggling a bit how to bring this topic to you. And then I was thinking about the course here, because basically here it's also an assembly, right? The host institution, the host, yeah, they provided us with the means, yeah, to be here together. And there was a selection that was done before you came here, right? Because many people registered, yeah, wanted to colonize the host, right? But there was just a sub-selection here that made it to the room, right? And then coming here, yeah, in terms of assembly, you need to disperse, you need to move, right? And I saw some of you, they came on a motorbike, right? Some of you came walking, some of you came together on a motorbike, right? So think about that. And then when you enter here into the room, there's the communication. So there's a lot of signals that are, from the hosts to you, but vice versa as well. So there's quite some chemistry going on, and that's a very important part of assembly. Then there is the food, yeah. So we communicate with each other. I think if the host institution decides not to provide us with lunch, I think we're going to have another communication here in the group, right? So that's also part of the whole assembly process. Then some people, they gather here. Some people prefer to be in the back, right? So that's, in terms of assembly, that's what they call compartmentalization. So people, they occupy different places in space here. And during this week, so there's also a temporal element here. You will see that sometimes these patterns, they change. Yeah, so, well, actually, so that was my lecture about assembly. No, let's have a look. So, but think about it in this way. Yeah, so these are the processes. And that's why I also put this picture here, right? There's many different members, some people communicate with each other, others don't. And when you talk about plant microbiome assembly, again, plants are surrounded by microbes from many different sources. Interestingly, for example, here, microbes in the atmosphere. I don't think that there's super interesting literature out there where they talk about microbes in aerosols, right? So those are microbes that can actually be transported or dispersed over very long distances. Then you have here, like I said, microbes in clouds and raindrops. And these microbes, at some point, they get into contact with the host, or they are recruited by the host. And a plant here, that's the host in this case. If you look at the above ground parts, there are the flowers. There's groups working on flower-associated microbiomes, so microbes only particularly located in the flowers. Then microbes that assemble on leaves here. I know there's a number of students here in the group that work on the phylosphere microbiome. So that's here. But you see this little insect here. So apart from airborne microbes there, microbes in the phylosphere can also assemble there through insects that are feeding onto the leaves, because also these insects, they carry microbes with them that then can colonize plant leaves. During my master's, which is quite a while ago, I looked at microbes associated with leaves, and particularly yeasts. And we looked at these microbes in the context together with aphids. Now, aphids, when they land on a leaf, and they deposit honeydew, the sugar compound, which has, again, a disturbance on the leaf microbiota because they externally provide a food source onto the leaves, which then, suddenly, shifts the microbiome to another composition. So the message here is that, it's not a static event. So microbial community compositions on plant parts is not static. It's a dynamic process, depending on these types of external factors, but also on plant developmental stage. I'm not going to talk about the impact of plant development on assembly today, but again, there is quite some literature out there which have looked at the different stages of plant development and how microbes actually change. And even, again, if you take a leaf and you would look at even the spatial distribution of microbes on a leaf, you will see that at different positions on that leaf, you get a different assembly of the microbiome. Okay, then if you look at the leaf microbiome can also be colonized by microbes, what they call vertical transmission. So when the seeds germinate and the cotyledons or the radical emerges, which means that microbes that are contained within the seed, they can go with or internally as an endophyte, they can go to the above plant parts. Okay, later on I will talk a little bit more about this. Then below ground, which is sort of like, we don't really see it, but again you have microbes in the bulk soil, which are also sometimes, through microbes in raindrops can be there. And these microbes, they are then assembling on plant roots and also inside root tissue. Then the seed-associated microbiomes. Are there people here that work on seed-associated microbiomes, so spermosphere microbiomes? Sorry?
Seed endophytes.
Seed endophytes. Okay, excellent. Good. So yes, indeed, there are a number of microbes inside seeds, onto seeds, and to make it even more complex, sometimes these seeds are eaten by, soil animals, which then, go through the guts, and then they, are deposited back, so they get some microbes even from the feeding. So you can see that assembly is super dynamic, and so we're trying to find, are there general principles in the assembly of microbes on plant surfaces. So the picture I just showed is actually super small scale, but it's interesting. There are a number of studies, and I think also here in India, where they do sort of like these biogeography type microbiome analysis. So you sample across a transect, and then you see, for example, on the same plant species, and this is an Arabidopsis plant, but you can do it for other plant species as well. So here they had a transect going from all the way north in Finland, Sweden, all the way down to Spain. And this area here, Spain, that's where Victor was born. So this is where Victor is from this area. Yeah. Myself, I'm here. Guillermo is somewhere here, and also Luisa, she grew up in Spain. So we have a Latin delegation here as well. We did not do any microbiome analysis on the four of us to see if this transect comes back. But the idea here is that every locality, going from all the way north here, all the way south, has a different environment and also has a different microbiome associated with that environment from which the plant can select. Okay? So, and that determines, and this composition here is determined by the local, physical chemical characteristics of that particular environment. And then again, the plant here selects microbes from that pool. And that's where you see the differences here in microbiome composition between locality 1 and locality 2. And again, there's the principal component analysis again, right? So what you see here, so again on the x-axis is that the two localities, they separate. And the percentage that is given there is usually the percentage that explains the variation between the two localities. The second axis here determines actually the variation that you find within each locality. That's the y-axis. And you can see here that this differences in for each of the localities. There is even again quite some variation in microbiome composition of plants sampled in that same locality. And there are a number of processes that drive these differences. One is called ecological drift, which is sort of like a stochastic process. If you look at the definition, which means birth and death of organisms, and also stochastic differences in growth rate of microbes. That determines why sometimes on a plant in the same locality you see another dynamics or another composition, I should say, for that same plant species, but still separating from the other locality. And that has to do with the distance and the dispersal limitation between one locality to the other. Now, what is also interesting in terms of these types of differences, they can also be driven by evolution within your microbiome at a specific locality. What is interesting is that element, so evolution, it's starting to emerge, but it's not sort of like studied in detail yet. So within plant-associated microbes, you can have mutations, which then would give that particular mutation an advantage in growth or advantage in the use of a substrate, which then gives that microorganisms a higher abundance in that microbiome community, right? Very interesting element here. It's emerging. There's not so much yet known about that. Yes, there is much known about evolution, experimental evolution in isolated cultures. But when it comes into a microbiome context, that is still to be explored. Okay, so what I'm going to do now, and I'm just going to go step by step. Some slides I will spend a bit more time, other, with less. But I'm going to go to the spermosphere, microbiome assembly, then rhizosphere, microbiome assembly, and then phylosphere, microbiome assembly. This picture I showed also during my lecture yesterday, right? There's a super nice paper from the group of Gabriele Berg in Austria. She does a lot of beautiful work also in terms of imaging. So they make slices of the plant material, and with specific markers or specific dyes, stains, you can stain microbes, and then you can actually look where are they located, right? And this is just a picture from one of their review papers where they showed, even the diversity in morphology of the different seeds that you have of plants. And then when you make cross sections here, you can see nicely that sometimes microbes are, located outside or in between plant cells and even inside cell tissue, right? This one, this is a very recent review paper where they brought together the spermosphere microbiome assembly. Again, it goes back to the picture I showed you earlier on, where we have here, let's start here, the flowers. Yeah, so that's where microbes are present. They are either on pollen or they are even in the maternal tissue of the flower, and then they can get internalized in different ways into the seeds, making so assembled in the endosphere of the seed. Then, of course, when the seed is formed, and again, like I said, here again there is an insect, that also visits these flowers. So also insects may, contribute to the microbiome composition eventually of the seeds. And then, of course, when the seed is produced by the plant, and if you go to wild plants, these seeds are then usually dropped into the soil top layer. And then when the seed is there, it starts interacting, of course, with microbes that are also present in the soil matrix. So they will colonize the external tissue of the seed. Some of them even go into the seed as well. And like I said earlier, there are also some animals that eat these seeds, poop them out again, right? And then you get, again, a different change. For seed companies, this is also quite interesting. So when seed companies, for crops produce seeds, of course, when they bring them into their company, there's a lot of treatments that they do on these seeds, especially for phytosanitary purposes. You have to clean these seeds from pathogens. So that in turn, so the seeds that you may be working with of crop species, they usually already went through some series of treatments which have impacted on the microbiome composition, mostly on the outside of the seeds. Okay, and then when you then go to the seeds and the seeds germinate to form a plant, you can have vertical transmission. So the vertical transmission means that it goes from the baby here up to the plant. Yeah, so the microbes associated with the seed, they will colonize the plant, both the upper surfaces and lower surfaces. But again, the plant is then also colonized also through horizontal acquisition of microbes that are basically in the surrounding environment. So what is interesting, so a group also in Austria from Angela Sesic's group, they thought, this whole seed microbiome, can we use it as a way to engineer microbiomes on seeds for the benefit of protecting plants? So what they have done, and that's shown in this cartoon, again in a paper by Birgit Mitter, where they have inoculated at the flower stage plants with, in this case, it's an endophyte, it's Paraburgtheria phytopermans, and they have inoculated the bacteria at the stage where the seeds are formed, and then these endophytes, they are basically internalized into the seed, and then later on when you plant these seeds, you can then find the endophyte back both in the roots, in the rhizosphere, and in the shoot endosphere. And they have visualized that here in this picture because the bacterial strain, and that's just as a proof of principle, they have provided the bacterium with a reporter gene, which has an enzyme which you can then, with the substrate, visualize. This was the GUS reporter that they used, and then they could nicely show that this bacterium was present in the endosperm of the seed. So I think they patented this technology as a way to introduce beneficial microbes into plant seeds for several purposes of plant growth and plant protection. Okay, let's move from the seeds to the rhizosphere and the endosphere. How many people are working on rhizosphere, endosphere, microbiomes, and roots here? Okay, good. That's a fair amount. I love this cartoon in terms of microbiome assembly. There is so much information in there. For every element in this slide, you can even have a separate lecture. That's what I love about this cartoon, because it is nicely comprehensive in a way. So rhizosphere and endosphere microbiome assembly. Of course, so here we have the plant root, which releases exudates, so that's the food source, and that reaches the start inoculum, so the seed bank, basically, and seed bank in terms of microbes. So you have a pool of microbes in the soil. Think again about the course here, the host institution and the selection. So the nutrients that are provided by the plant root, so depending on the composition of these root exudates, only a fraction of this microbiome, this soil microbiome, can utilize these substrates either through active transport of these substrates into the cell or by degrading these compounds into more easily, utilizable substrates. So from here on, you get a sub-selection of microbes that is able to respond to the signals, the chemical signals that they get from the plant. This sub-selection here, and that's the panel here, of course, they start to interact. And so they start, multiplying, they start to produce toxins, they start to activate defense systems because their neighbor is producing things that will inhibit their growth, so they have to defend themselves. So there's also quite some what they call antibiotic resistance genes at this stage that are activated. Also CRISPR-related defense systems to defend themselves against bacteriophages in that population. And then also at the same time, there are quorum sensing systems activated. Are people familiar here with quorum sensing? Yeah? So that's cell density dependent regulation of gene expression. So the microbes, they need to reach a certain quorum, certain cell density before they express specific genes. So that is also an important element of the competition that is going on. And then two-component systems. Are people familiar with two-component systems? No? So two-component systems is when microbes receive a signal from the environment, and they receive that through a sensor kinase, which is usually located, at the outer membrane. And then when that sensor kinase receives the signal, it is autophosphorylated, and then transmits that signal inside the cell to what they call a response regulator. Yeah? And the response regulator then in turn activates a number of genes. So we have two components. We have a sensor kinase in the membrane and a response regulator inside the cell. Right? So that's the system. And that activates usually, so two component systems, they activate a number of traits. Production of secondary metabolites, motility, etc. So here also, that's the next stage. So, this community that is selected through the substrates here, they then, in this nutrient gradient, they start moving towards the plant root through chemotaxis. So they sense, the chemistry of the plant. They want to go there. And then there's another flagella, motility proteins, they are all activated. So they start moving and they start multiplying. So you see that the population is growing again, but the composition is still a sub-selection of this initial soil inoculum, right? Good, so they start, approaching the canteen, right, for the food. And then they start to aggregate here. Again, forming these biofilms. There's a lot of, again, communication and signaling going on here. Two-component, three-component, multi-component systems. Again, there's a lot to tell about that, but I will leave that here. There is some other secondary messenger, so the cyclic di-AMP, and then, of course, also the plant compounds that are there that provide the substrate, but some plant components are also a signal. What is interesting here is so also at this stage, but also when some microbes, they also would like to go inside the root system. And going inside the root system again is sort of like a next filter of selection in the assembly. So some microbes are able to do that, others are not. So they need to have specific traits to colonize the inside tissue. So I'll walk you through here. So the roots, they are producing a lot of compounds that are also toxic to the microbe. And one of these compounds are reactive oxygen species, like hydrogen peroxide, right? And several of the endophytes, they have enzymes that detoxify these reactive oxygen species. Are people familiar with the enzymes that microbes use to detoxify reactive oxygen species? Yeah, superoxide dismutase, right? Catalase, peroxidase. Yeah, good. Yeah, sorry, I sometimes when you're here, it's difficult sometimes to hear you because there's an air conditioner, you know. And anyway, good. That's another issue. So, so that's one thing indeed. So that's an important trait of microbes that colonize the endophytic space. And then here it says lytic with an arrow down. So many microbes, they produce these cell wall degrading enzymes. But when these cell wall degrading enzymes are expressed at a high level, the plant will probably recognize these microbes as a pathogen. So for microbes to endophytically colonize these plants, they need to sort of like down-regulate the production of these lytic enzymes to avoid being recognized. A lot of transcriptional regulators, MUMPs, that are the molecular associated or microbial associated molecular patterns, are down because that will also, be them for the plant to signal that they are recognized as a pathogen. Secretion systems are down, effectors are down, and then we have the preparados. I will not talk about the preparados because I think one of the groups here who was selected for the Asaf Levi paper. In the Asaf Levi paper, there's some information on preparados. So it's a cliffhanger for next week, Tuesday, right? Okay, so and then of course, among these microbes that are selected by the plants, of course, there are also pathogens that are recognized either through MUMPs or effectors as pathogens, and then the plant responds accordingly. So this is all, and later on in the slides, I will go again a bit more into detail. This is all when the plant is healthy. Imagine, and I know there's some PhD students here working on, suppose that a plant is infected by a pathogen or by an insect or by a nematode, right? Then, of course, you can imagine what happens, and there's an example here given here. So when there is a stress response here, sometimes, plants, they produce the coumarin scopolamine, and scopolamine by itself, will change the exudate composition, and that will immediately, have an impact on the selection of the microbes from the start inoculum here. Again, emphasizing that this is a super dynamic process. It's not static. Is this slide fully clear to everybody? There's a lot of theory behind it, but I think it's laid out beautifully. All the different elements of what selection from the plant. Microbe interactions. Some people spend their whole scientific career on biofilm formation only, right? So that's only this element here. There's a lot of interesting stuff about the interactions when it comes to all different types of metabolites or secretion systems. So beautiful cartoon to basically bring all the elements of assembly together. So let's move above ground, the phylosphere. Assembly, this is actually a classic. I love this approach to just visualize the distribution of microbes on a leaf surface. So what you do is that you take the leaf and you print it onto an agar plate. And depending on the agar plate that you use, you can, of course, filter out specific microbes and then visualize where are they located on the leaves. And this is one of the examples how it looks like. Again, it's a lot of cartoons, but I think, it brings back, together visually the important elements of microbiome assembly. So phylosphere, like I said earlier, it can be colonized vertically through the seed, but also externally through the environment, like here with the climate. The plant species itself, so the genotype of the plant, and also even cultivars within a given plant species, may also have an effect on the assembly. Agricultural management, the example I gave from the honeydew, what we actually looked at in that project, in that master project, was if specific fungicides are used in management, so you basically eliminate fungi on your leaf, it will have an impact on how this microbiome of your leaf is starting to be composed. Seeds we just talked about the vertical transmission. Of course, soil type and properties will have an impact also on exudates released by plant leaves. Herbivores and phytopathogens. We talked about the insects. I know that there are people here working on the impact of specific leaf pathogens on microbiome assembly, right? So, which pathogens are you using? Okay, right. Right, yes. I think there's more, right? There's also people working on bacterial pathogens, right? Yeah, what is quite interesting in that topic, as you know, if you look at pathogens, there's a number of, you have necrotrophic pathogens that basically destroy tissue from outside. And there will be a lot of nutrients released, which will have an impact on the assembly. And then you have the more biotrophic, hemibiotropic, that first penetrate tissue and then grow inside the plant tissue. You can imagine that even at that level, you will have a different impact on microbiome composition. And I did not even talk about, up or down in the leaf, because on top of the leaf, there's a lot of UV radiation, right? So microbes are super sensitive to that. Whereas on the other side of the leaf, your microbiome might actually be different again, right? Okay. Yeah, one other, advice for recommendation, super nice paper, but it's more an evolutionary type paper, focused on, again, microbiome assembly and how the host actually keeps the microbiome on a leash. So how the host select. And this paper by Kevin Foster, it was published in 2017 in Nature. And he and his colleagues, they compared microbiome assembly of humans, of corals, and of plants. And it's really nice reading, especially when they start to talk about, the similarities, these cross-kingdom similarities in traits of the host that select microbes, on their surfaces and inside tissue. So both in the humans and in the corals, you have these mucus layers. And in plants, these are consisting of arabinogalactans, right? So that's the plant sort of like version of the mucus layer that you find for corals and for humans. And then the green arrows actually mean a positive effect on the assembly, and the red arrows are actually, describing traits of the host that repel or deter or inhibit microbes with the assembly. So you have the glycoproteins, you have the immunoglobulins, and then you have a number of antimicrobial compounds. And here again are the acidification, that's usually organic acids that are released from the host, also as a negative effect, and also the reactive oxygen species. And then, of course, the nutrients, that attract, a subset of the microbes. Have a read through, but what is interesting in this paper, so assembly, they try to, bring some structure in the processes that determine assembly. Regardless if you talk about seeds or phytosphere or rhizosphere for plants, they talk about three processes here. So it's the microbe to host interaction. And there, of course, there is the issue of the diverse microbiome. So you can have assembly of beneficials, of neutral, but also of pathogenic microbes. Then there is the host to microbe signaling. And that's in the paper, they talk about this in terms of the importance of host control. And in the cartoon of Trivedi paper, we talked about many elements already here, but I'm just going to repeat them again. So, of course, you have the immigration. So, microbes moving onto the plant surface. And you have compartmentalization. And compartmentalization can be in space. So you have microbes that prefer to colonize the root tips and microbes that prefer to colonize older. So that's a compartment. But you have also microbes that stay on the surface and microbes that can go inside. So they compartmentalize on the plant. So that's, and then you have the vertical transmission, like we talked about, from seeds to plants in this case. And one point I did not talk about yet is so-called priority effects. Very simple. Who is there first, right? Who is first at the buffet? And that sometimes determines also the success rate of a microbe to be there. And when you talk about applications of microbes for specific plant growth or disease control, priority effects is one of the major issues. So when do you introduce a microbe? If you introduce the microbe too late, it has to establish on that plant surface in competition with all the microbes that are already there at the buffet, right? So that's why many times they try to bring the microbes into the system as early as possible. When you talk about leaves, when a leaf just unfolds, there's very few microbes there, and that would be a good moment. And that's why many applications also start applying microbes to the seeds. So when the seedling emerges, those microbes that you want to be there are there first. Then in the dispersal invasiveness, we talked about that, and then the plant also does what they call monitoring and targeting. And monitoring means what I talked about. Is this beneficial? Is this neutral? Or am I dealing with a pathogen? And they have many different recognition systems to basically filter that type of signal information. And then in terms of targeting means that a plant releases specific substrates that can only be used by a subset of the seed bank of the soil microbial inoculum or by the environmental microbiome. And an example that they give in the paper is glycans. These are quite complex carbohydrates, and there's not so many within the microbiome. There's not so many phyla, genera, species that can use that. But for example, there is one group, the Bacteroidetes, that is very able to utilize these complex substrates because they have the enzymes to degrade these compounds. And then microbe to microbe. So, like I said, there's a subset selected, and these microbes are then together, and they start competing, cooperating, so surviving in the microbial jungle, and they start to co-evolve into the assembly. Again, it's exactly the same slide as I showed before, but then more in a cartoon, like. And what in this paper, what they did is so you have plant to microbe signaling, microbe to plant signaling, and microbe to microbe. And they put some chemistry here. So the chemistry that is underneath, which we talked about already a bit. So the carbon fluxes, reactive oxygen species, organic acids. Here, we did not talk about that yet. Again, there's so much information here, but auxins, which are plant growth promoting hormones, that also some microbes produce. Super interesting. And then here, this whole story about quorum sensing, two-component regulation, but also communication through volatiles. And that is shown actually the type of interactions that can happen in this assembly between microbes when they move towards the plant assemble on the plant, and then still keep on interacting. So you have contact-dependent interaction, type 6 secretion. So this morning I thought, shall I talk a bit more about type 6, type 4, type 3? And then I got lost, and I thought, no, I'm not going to talk about it. But there is super nice information there. So type 6 secretion, it's quite an interesting process where microbes inject toxin into their neighboring organism. Of course, competition for iron is a classic example. I did my PhD thesis on competition for iron, where microbes produce these iron scavenging compounds called siderophores, and then you can compete with other microbes, with pathogens, and that all depends on the specificity of the siderophore. Because if you have a very specific one which cannot be utilized by the other one, you have a competitive advantage. And some microbes in iron competition, for example, they have receptors by which they can actually, gather, iron from other siderophores, which again gives them a competitive advantage. I think the contact-dependent antagonism in the practicals today, we will do so with the microbes that we isolated from the wheat. We will do some, interaction on a plate, right? Antagonism assays. But of course, there's also cooperative interactions where microbes work together. They are in multi-species biofilms, quorum sensing. They talk to each other, either through quorum sensing molecules, or sometimes through volatile organic compounds. And the interesting part of volatiles is that you can communicate with each other at a longer distance, because these compounds, they diffuse over a long distance. And then last type of interaction in this cartoon is nutrient exchange, which means that sometimes microbes cannot utilize a specific substrate, but their neighbor can. And when the neighbor uses that substrate, the degradation products of that substrate can then be utilized by the other organisms. So that's also a way of basically cooperation. Yeah, good. So this is all when plants are, growing well, not stressed at all. And then real life kicks in with a number of stresses that they are exposed to, which means abiotic stress. So we have drought, salinity, UV, and then also on this side the biotic stress with fungi, insects, nematodes, and plants by themselves. So without the microbes being there, also have, of course, defense systems or attractive systems to bring microbes in or to repel, and that's depicted in this picture here. So, for example, tolerance to drought stress, with the osmoregulation in roots with prolines, then the coumarins, that is a compound that plant uses to get iron. And then you also have a number of organic acids which plant roots exude to get, for example, solubilization of phosphate. And then on the other side, of course, these types of signals are not only used by beneficial microbes, but they are also used by pathogens. They are also present in the system. And then plants have sort of like defensive compounds, and that's depicted here. So phenolic compounds that inhibit soil-borne pathogens, DMDS and DMPS, those are the sulfurous compounds released by plants, dimethyl disulfide, dimethyl trisulfide, which have quite a broad spectrum activity against many pathogens. And then, an interesting part, the terpenes. If you do an anti-smash analysis with your genome sequences of your microbes, the hits that you get back, you will find a lot of terpenes. And there's Paulina Garbeva in my group. She's working on terpenes. And terpenes are not only produced by plants, they are also produced by microbes and many other organisms. And Paulina, actually, she proposed that terpenes might actually be the language by which, organisms from different kingdoms communicate with each other. It's an interesting thought. So, okay, good. Then drought. Now drought, again, just to illustrate the complexity in this figure, drought, of course, can have a direct impact on your soil texture. And when the soil texture changes, also the water content in the soil changes, that in and itself, without the plant even being there, will have an impact on your seed bank, on the soil microbial community from which the plant will start to select microbes. That's one thing. The drought may also have an impact directly on the microbes. Some microbes are much more drought tolerant than other microbes. So that will also have an impact. And then, of course, it will have an impact on the plant, on the chemistry, but also on the root architecture, right? So, again, illustrating the complexity here that when a stress comes into the picture, that the whole assembly of the microbes on these plants may change dramatically. And that's also, I will not go here, but this is also what is depicted in this picture here, where you have, the increased stress resistance. So when plants start to recruit beneficial microbes upon the stress, or when they induce susceptibility, when pathogens start to, when a plant is stressed, are, in favor of colonizing the plant. Okay, for me, a couple of more examples before we go to a closure. For me, this paper is sort of like one of the first papers I thought was super nice. This is one of the first papers from Rudrapa, from Harsh Bai's group. And Harsh, his group, they work on basically chemical communication between plants and microbes, so which chemistry is involved in recruiting microbes. And they did a beautiful study where they exposed tomato plants on the leaf with a bacterial pathogen Pseudomonas syringae, and then on the roots. So they had a soil where, in this case, they inoculated a Bacillus subtilis strain. The strain is FB17. That's not so relevant. So here you have the pictures of, this is a staining with a dye of the bacillus on the roots, and the leaves are treated with BC3000, that's Pseudomonas syringae, part of our tomato, which is on that. Then you have a non-treated control, then you have a mock, and the mock is where they infiltrated water into the leaves, so not the pathogen, but just the water. And this NPS 3121, that's a non-pathogenic Pseudomonas syringae. And what they showed in this assay is that when leaves of tomato are infected with Pseudomonas syringae, you get a significant increase here in the density of your bacillus, the cell density you find on the tomato roots, as compared to the control, the mock inoculated, and the non-pathogenic Pseudomonas syringae. So you need an infection there on the leaves to actually, enhance the density of your Bacillus subtilis on the roots. They did a lot of more interesting stuff here. So the story here, so like I said, Pseudomonas syringae, it infects the leaf surface. You get a signal going down. And what they discovered is that through a transporter, the plant starts to release more malic acid. And it's exactly this malic acid that, causes the enhancement, the recruitment of this bacillus strain. So through chemotaxis, it colonizes the roots. And by colonizing the roots, it gives something back to the plant. It induces a resistance response in the plant, which means that defense responses are activated and subsequent infections of the Pseudomonas syringae are suppressed. That's sort of like the whole loop. And there was quite some experiments in terms of validation. So they also had a paper where they used a mutant in this malate transporter. and that was not able to actually trigger this whole chain of events. So with that, so that was one of the first examples, and that was a recent review paper. And I'm going to share the slides so you can look at this in just easy. How do plants modulate the root microbiome through chemistry, by root chemistry? I mean, again, 3 slides with tables. No need to read the details now. I'm just going to highlight a few. So the first one here is what that's work from the Utrecht group in the Netherlands, from Corné Pieters's group and Roland Berendsen and Peter Bakker, where they actually looked at what they call the soil-borne legacy. So when plants, they also have the soil-borne supremacy. They even have, so they are totally fond of Jason Bourne and this whole sequel of movies. But what they did is they infected Arabidopsis plants with Peronospora on the leaf, and then they looked on the roots which microbes are differentially abundant, right? What we learned in the last 2 days already. And then what is the effect of the differential abundance on the phenotype? And this paper here, that's one of the papers that also one of the groups here will present next week Tuesday. So that's why I highlighted this one. Then we have work from my own group where root infections with Rhizoctonia solani caused a shift in the microbiome composition on roots of sugar beet. And it's interesting, so what we saw is a lot of bacteria belonging to the group of Oxalobacteriaceae. And I think the mycologists here or the plant pathologists may know that many plant pathogenic fungi produce oxalic acid by themselves as a virulence factor, right? So the oxalic acid has many different properties which help the fungi to infect the plant tissue. And it's interesting to see that when we inoculated these plants with Rhizoctonia solani, which also produces oxalic acid, you get a big enrichment of these microbes that can utilize this substrate. So that was one. The other thing here for people working on insects, there's some really nice work also done where they infect the leaves and then they look for the recruitment again of a Bacillus subtilis again, which then has an impact on a root pathogen Ralstonia. Here at the bottom, I think we talked about that. So iron deficiency and colonization by PGPR. So that's work also from the Utrecht group where they inoculated leaves. You see increased coumarin production, and these coumarins, it turns out that they have also an antimicrobial effect, but a differential microbial effect on microbes that are there. So they had a higher activity against pathogens, and whereas beneficial microbes were not affected. So there you get some nice. And last but not least, again, there's so much interesting work out there, but this is a nice summary. Plants that produce benzoxanoids, which is also an important class of compounds released by plant roots and important in assembly. And then also work by Anne Osborne's group where they looked at the release of triterpenes and how that impacts on microbiome assembly. Lots of information, I understand, but so the bigger picture here is that plants release a lot of compounds that have a selective effect in the assembly process. And depending on the stress factors that plants are exposed with, they change their, root exudate composition. And then basically, the cry for help has been mentioned also in below ground contexts, that plants, when they are attacked, they change their chemistry to recruit beneficial microbes for subsequent protection. That's the concept and theory. One last example. That's a PhD group, student in the group of Paulina Gaveva in my department. We are very interested in volatile compounds released by plants because they can diffuse over quite a distance. and we developed an experimental system here. So I'll walk you through it. So you have a little jar here, plants growing in there, and you can sterilize this if you'd like. And then it's connected here to a tube which captures the volatiles. So you later on you can measure which volatiles are released. And on this side, there is a little space here, and then you have a tube which you can put soil in. And you can sterilize these soils, so you can use natural soil in your system. And then on the other hand, you have, of course, the control where you only have the jar without the plant. You can also expose this plant to stress or not to stress. And what Mohammed did is he uses a sterile soil and he inoculates a soil microbiome suspension here in the center. And what he does then, he incubates these plants and he starts to look for which microbes from this inoculum source here start to move there or preferentially start to move towards the plant. Right? And this is how it looks in real life. And the nice thing of this system is that on this side with the volatiles, we can do metabolomic analysis. So which volatiles are released by the plant. You can do different developmental stages, or you can, like I said, you can expose the plants to stress. And then on this side here, you can sample this part of the tube and also this part, and you can do 16S amplicon analysis or metagenome analysis to see, which microbes move here and which microbes move there, because that will be your sort of like your control. So that's what Mohammed did. So this is the chemistry. It's not so important details, but you can clearly see that when plants are eaten above ground by an insect, the root chemistry with the volatiles changed. And you even could pinpoint, the compounds that are, more abundant when plants are eaten. So again, dimethyl disulfide I mentioned before. And then also with the microbes, so the compositional plot, right, that we also did in the practicals yesterday, 16S amplicon sequencing, he could see that this is the sterile strand. A healthy plant is here, and when plants are attacked by an insect, the population starts to separate up. Okay? And then you can do what we did yesterday, you can do the differential abundance analysis, right? Which microbes explain the separation on these axes, right? And then this is a bar plot at genus level. But you can also take that soil, like we did in the practical, you can take that soil, do the plating, and then cross-link these microbes again here. So then you have a collection of microbes that are attracted by plants when they are under stress, and you can test those microbes again back. If you apply them to the plants, do they provide protection against this insect stress, right? So that's a bit of the PhD project that Mohammed is working on. This slide, too much information. We have the same, all together in the group, TMI, we call it, too much information. But this is a full summary of what I just talked about. And I will share the slides. You can just go through it and then, have a look at it, right? So I think with this, I will close. That's okay. Thank you. Are there any questions following assembly? Yeah. I'm going to stand here.
So if there is any competition, nutrient exchange or coming to the vertical transmission from seeds to flower. So here I can say that the microbe transmit only the non-competitive microbes or the cooperative group only are getting transmitted. If other factors are affecting them, then we can say that only the microbes which are non-competitive or which are cooperative that move from seeds to flowers?
I don't know. I don't know if, let's say, cooperative organisms preferentially colonize, or assemble, or whether you have to have competition between the two, or the ones that are more competitive. I think it's probably, a balance of both. Yeah, yeah. Okay.
We were saying about the mechanism of root exudate of the volatile compounds, root
Right.
exudation pattern.
Yeah.
So can you give me information about seed exudates also?
Seed exudates, yeah. That's that, yeah. So during the imbibition process, right, that there's a lot of nutrients released. There's some nice reviews by Eric Nelson and also by Mathieu Barrette on the whole seed chemistry, spermosphere chemistry, and how that impacts on microbiome assembly. So also there, at the seed stage, especially during the imbibition, you get this, boost of carbon sources, vitamins, or maybe not vitamins, but amino acids, that are released, and that, of course, immediately attract microbes to the buffet. So there is some information available also on chemistry of seeds. Absolutely, sure, absolutely, yeah, yeah, because there's a lot of sort of like what they call pre-emergence damping of diseases, right? That like to attack the seeds at that point. Yeah. Okay. Sorry. Yeah.
In your very first slide, I saw the arrow mark vice versa also from seedling to root.
Yeah.
So, like the question is like we have seen about the vertical transmission.
Yeah.
So, how about the transmission from flowers? Like whether it all root gets sprayed in flowers.
Yeah.
There might be also the migration or transmission from flowers vice versa.
Yeah. So that's a good question. So when you have that, probably the microbes, they need to enter the vascular system, right, to move downwards. I think there are some examples in literature, but the CD-ROM is not there at that paper. So okay, but there is some information on, but not so much, I must say. Yeah. Yeah. Thank you.
So kind of the mechanisms where by which endophytes interact with the plants.
Yeah.
So in that case, there was a point that the hydrolytic enzymes produced by the endophytes may, like because the hydrolytic enzymes are produced, the endophytes may not recruit them. But sir, when we isolate the endophytes and we see certain endophytes producing hydrolytic enzymes,
Sure.
that will show some antagonistic effect over the pathogens of that particular plant. So, what is the relation in that case?
Yeah. So yeah, the cell wall degrading enzymes. So what basically the proposition or the literature says is that to become an endophyte, if they express cell wall degrading enzymes at a maximum level, the plant will perceive that organism as being a pathogen. So for endophytes, they still produce the cell wall degrading enzymes, but to a level that the plant perceives these microbes not so much as a pathogen. That's basically, so yes, if you test these endophytes, you will see that on plate, right? If you do these, the halo assays, whatever, yeah, you will see that they produce the cell wall degrading enzyme. In situ, for these endophytes, they are probably at a much lower production than when you use a pathogenic microbe. Yeah, yeah. Victor. And well, they are not only those insects, there are other determinants like for example, the type specific system and the effect of protein.
Yeah.
So many of these beneficial microbes don't have the effect, although they have the type specific system. Yeah. You know the type specific system that affects.
Yeah. So that was. Maybe, yeah. Yeah. Yeah, so just to, again, come up to that, so there was this sort of like list of traits that was mentioned there to become endophytic, yeah, lytic enzymes down. There was the mumps and the effectors, and so that was, it's not just one trait, yeah, that it's a complexity of traits and the regulation of those traits, yeah. Good.
Some more questions? Excellent lecture. Anybody?
I think more questions will come during the day, Kumar. Yeah, so if things come up, you know, just ask, okay? Good. So, Kumar, the coffee is again at 11 o'clock? Or 10:30? Okay. Then maybe, Vic, it's a good moment, you know, to hand out the presents that we bought for Ramakrishnan, the team, right? It's, yeah? Okay. I'm looking at Javed, and so where did we leave the, I think, yeah, sorry? Yeah, exactly, yeah. No, sure. But you have seen, I think, in the lectures, and that's what I try to bring out, is that, you know, all the different elements, right, for microbes to assemble on a plant surface, or, you know, even if another host, you know, there's so many factors. So to really understand, you know, the major drivers that determine that assembly is super difficult. It's super difficult. So you remember in my first lecture yesterday, I said, you know, understanding the major mechanisms in assembly, yeah, predicting assembly. If we can get to that point, of course, you can much easier engineer a microbiome for a particular trait. It's, but you see the complexity there. Yeah. Okay. Yeah. Good. Do the endophytes? If yeah, that's a good question. So yeah, to be honest, I don't know the answer to that. There are some true endophytes, and there are endophytes that come and go. And then when the plant ages and parts of the root system, for example, dies off, I think then they will start to exhibit a different type of expression patterns by using, you know, their host, which has been there for a while, as a substrate. Absolutely. Yeah, 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 yeah.
The role of endophytes and structure, the role of endophytes and structure of the stage from the vegetative stage to the reproductive.
You mean the dynamics of endophytes? Yeah.
So it will decide the plant structure from the vegetative stage to reproductive.
I don't know the answer. It's a yeah, sorry.
So we are applying some fertilizer, for example, phosphatic fertilizer. We can able to make the plant to vegetative to reproductive stage. Or when the plant is expressing sensing the stresses, it will quickly come into the reproductive system.
Right.
Likewise, some innovate which is present in that particular condition, stress
Right.
conditions of which will decide the plant to switch over from the reproductive stage, or even delaying the senescence of
All right. Okay.
microbiome for delaying the senescence of
Right.
to withdraw under the particular system.
Yeah. 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 I understand the question, but I don't know the answer. That's. I wish I would. Yeah. But it's true that certain stresses in plants, you will see that they indeed will accelerate the development and the seed set. And that, of course, in turn will also have an impact on the composition of your endophytic community, of your phylosphere, rhizosphere community. So you have experience, you have data on that?
Yeah.
Okay, can you
We
share?
did the experiment in soybean bradyrhizobane interaction.
Okay,
We applied the stress in the field and we started imaging the field with the different treatment. And the plant which is applied with bradyrhizobane, which is fixing nitrogen. So there is a delay in
okay.
this has been captured through images.
So what you're saying is that the symbiont, by inoculating the symbiont, it delays,
Delay
the senescence. All right.
when we apply
When you apply the stress. Oh, super. Yeah. Okay. Thank you.
Okay, thank you, sir, for the presentation. Okay, my question is, we've seen that there is cross-sectional interaction in the soil, the rhizosphere, the hemisphere, and so on. So now, when you want to provide a biological solution, so that you are fixing a particular problem, you need to solve a microorganism.
Yeah.
So what should we fix? How should we do to be sure that the microorganism we are providing to the soil will have the target solution we are, because there are too much interaction. We've seen that the interaction between the root and the microbes, interaction between microbes. So now, how should we be sure that, okay, this biological solution will just target this microbe, have the solution we need?
Yeah. I understand your question. So let me say first, there is no silver bullet. Let's start with that. Secondly, and that's why I think the field is, so yesterday I talked about transplantation and about single microbes, and that's why the field I think is moving now to consortia. The fancy term now is synthetic communities, where you actually provide microbes, sort of like an assemblage of different microbes that can have then different functions in that particular system. That's one thing. The second is when you apply microbes, they need to be at the right time and the right place for the particular problem that you want to address, right? So if you want to address pre-emergence damping of diseases, yeah, you need to make sure that your microbes are delivered either as a single or as a consortium when the seed is there. Whether they survive later on in the system or are still there, that's, much less relevant. But if you're dealing with, for example, diseases much later on in plant development, then you're going to expect quite a bit from your microbe so they need to survive. They need to still be there at a density where they can exert their beneficial effect. So, and that's been a major challenge. So one of the things that we have not talked about, but I will talk about that next week, is you can basically forget about introducing microbes, but instead, stimulate a fraction, so the beneficial fraction of your microbiome that is already in the system, by applying specific, precursors or substrates that selectively enrich the microbe at the time when they are needed. Super interesting development there as well. And yeah, I don't know yet, or you can even have a combination of both. where you provide a substrate with a microbe or a microbial consortium to basically kickstart the system. Yeah. But the question you have is a super good question. It addresses actually the difficulty of translating all this knowledge into an effective management practice. So, yeah.
Any more interactions? Yeah, please stand up. It's louder. Mic, please. Online people are not listening.
So I wanted to ask that if there is an immunocompromised plant, might be due to some environment or maybe some, the chances of these epiphytes or endophytes becoming pathogenic to that plant
Poof.
in
I see a new project arising.
that because you immunocompromised humans, you see various types of infections which do not infect the human.
Absolutely, yeah.
Are there chances?
My answer would be I think there will be, but this is a super good, super nice idea. Yeah, write a project on that. Yeah. No, that's yeah. Of course, I also talked about the good, the bad, and the ugly yesterday, right? And when some plants are immunocompromised, they can be colonized by opportunistic human pathogens. That's quite a big phenomenon here. But let's say I don't think mechanistically there has been a lot of work done on this. Immunocompromised plants, there are a number of studies, of course, with mutants of Arabidopsis which are immunocompromised in signal transduction pathways, how that impacts on the microbiome. But then, again, the extra dimension that you say, are they preferentially colonized by opportunistic or true pathogens? That is an interesting question to look at. Maybe there is some literature out there which I missed. That's well possible. But it's a super interesting idea. Yeah. Thank you.
Anybody from this side?
Good. Victor, shall we? Thanks a lot. For us, I like this a lot. So also yesterday during the practicals, what we saw is that based on the questions that you ask us, for us that's always sort of like a signal that certain things landed. So that's important. So and I see with the questions here exactly the same. So I'm super happy that these questions come up and even taking it to the next level. So super, yeah. Okay, good. We have some presents, right, for the group, and then we can share that with everybody again, right, Vic? Well, that's up to the group, of course. Vic, please go ahead.
Well, we brought more cookies from the Netherlands. So, and yeah, we would like to, to give it to outside.
Yeah, sure. So it's for the team that did a fantastic job here in organizing everything in the lab, right? Ramakrishnan, Kumar. Yeah. We keep on feeding you with cookies. Now, I have to say, the waffles that the ones that we had yesterday, they have been in the cold room, so they were a bit, chewy. We kept these outside, so that's the contrast analysis. So we'll see how that works out, okay? Okay, let's enjoy the break, okay?
I take this opportunity to thank Professor Bill. He has given subject clarity and complex problems, kept all of us very attentive to research. Thank you again.
You're welcome. Thank you.
Thank you very much.
So with that, we concluded the first lecture. We'll have a tea break now. And we'll come back at 11 o'clock.
Then we will have another lecture by Victor, Microbiome Technologies and Analysis. So that's going to take another 1 hour. But my only request to all of you when the presentation is going on, try to avoid movements within the hall unless it is really needed, and pay attention and try to be more interactive. So I think you would agree with me that some excellent questions came from some students from the backside, and don't hesitate to ask questions. It's not questioning the speaker, it is rather creating an opportunity for more interaction. So that is the philosophy. So let's break now. Yeah.
Interaction always takes time to
university or an agriculture research institute, you will be interested in creating technologies for the farm. This is going to be an important lecture for us to know what kind of technologies to be developed using the basic information which we gain from Microbiomes. Also, we will be continuing another part, analysis, which are supposed to be.
Thank you. Okay. Oh, great. Yeah, I think now, yeah, you can hear me, right? Also, our attendants online, the participants online can hear? Yeah, I guess so with this. Okay. Well, I got this nice microphone today, so then I think it's going to be better because I move a lot. Well, after the amazing lecture from Jos, I'm gonna change to another topic. So we are gonna be talking about, well, microbiome technologies and analysis too, and we will be focusing mainly on genome assembly, not microbiome assembly, but genome assembly and, well, phylogeny and, well, some other tools. Okay, so let's get started. So, well, we have seen already quite a bit from the program. So we have been here culturing bacteria, looking at the assembly of bacteria in the lectures of Jos. Also, we have looked at the taxonomy and the how to analyze that data. But, well, later this week or next week, we will talk about metagenomics. And, well, yesterday we also talked about differential abundance, how to do it, and so on. And today we will work in the lab. We will look at the bacterial colonies that we got from the rhizosphere of wheat plants. You remember that we did the culturing? Exactly. So, well, yesterday the colonies were already growing quite well. So I'm guessing that today we will have, yeah, nice colonies popping up. So then that's why today I'm following up on the lectures, on the lecture from Jos. So we are going to focus in, okay, so we cultivate the bacteria, and now what? Okay. Yes. So this is was prepared. This figure was prepared by Luisa. So you will see today again in the afternoon. So you grow your bacteria, and then you re-isolate them, and then you do your test. Depending on what you are working, you will be, yeah, most likely doing plant growth promotion experiments. You will see if, yeah, your bacteria can promote the growth of the plant or not, or any other phenotype that you are interested in. Also, antagonistic activities. You could see if your bacteria is producing any diffusible compound, yeah, that it's involved in the antagonistic activity against plant pathogens. Not only diffusible, as Jos was also mentioning in his lecture, you can also look at volatile compounds. But then once you get all of this information, yeah, well, you are interested in going a bit further. Yeah, you are interested more in, okay, so which genes are involved in these phenotypes that I am seeing, right? So for that, we do genome sequencing, right? So we sequence the genome of the bacteria, and then we do genome mining. So now I'm gonna go a bit back, and then I'm going to show you what to do when you get a genome sequence, okay? You get your FASTQ files, like yesterday we saw, right? But then how to analyze and how to interpret the data, how to assemble the genome, how to see what kind of bacteria you got, the phylogeny, and so on, we are gonna see today. Okay, like always, when you go down into the process, there are so many tools available. So I'm going to be mentioning some of them. In the afternoon, Luisa and Guillermo will be mentioning some others. So if you have any question, please raise your hand. Okay? Good. Well, these are going to be the learning goals today. So we are going to go a bit back again, and we will talk about quality control and data processing. We will talk about genome assembly, yeah? But what we are saying today, it's also applicable to metagenomes, yeah? To metagenome assembly. So remember, shotgun metagenomics, when you sequence all bacteria or all microorganisms present in a particular environment. We will also look at alignments, yeah? How to do genome alignments or sequence alignments. And then tomorrow, I think, because today we will not have time, we will look at annotation tools and some snapshots on comparative genomics. Okay? This is going to be the outline today. So, well, I'm not going to repeat again, but basically we are going to look at two different methods for genome assembly, two classical ones. So we have the overlap layout consensus and string graphs, or the Bruin graphs. We will look at the methods for genome assembly or sequence assembly, alignment, sorry, global, local, and the mix of both local. And we will see some tools for phylogeny. Here I put you some key papers that, again, we don't want to overwhelm you with a lot of literature, as just said this morning, but it's more to give you a guideline, yeah, just in case any of you is more interested in the topic that we are showing you here. Okay, let's get started. When you get your genome or your metagenome, you get several, right? You get several samples. So the first step, yeah, that it's done by the company, usually, it's the multiplexing, okay? So you have the multiplexing and the demultiplexing, okay? What is that? You get your samples. Every green line that you see here, it's a genome, or, yeah, well, a metagenome, okay? So then you have sample A and sample B. So we have an index that is a short sequence that is unique for each of the samples. And you can see here in different colors, yellow, purple, and then green is the DNA sequence. So what you do, or what the company does, they pull together, yeah, both indexes, the index for the sample A and the index for the sample B. Okay? And then they put all the samples together. That's a very scary moment, because you take all your samples or your treatments and then you pull them in the same Eppendorf tube, and then they put it in this machine, the sequencer, and then you get a mixture of reads. Yeah? You get a mixture of reads that contains index from sample A and index from sample B. Yeah? So the first thing that you need to do now is to demultiplex and get back the order in your samples. So sample A, so the yellow one, should go with sample A, and sample B should go with sample B. Clear for everybody? Yes? Thumbs up? Yes. There was a thumb like this? No? Okay, good. Okay, so there are some tools to do this, like Sabre or IDEMAX, but these are good news, because normally this is performed by the sequencing company. So you really don't have to take care of this process normally, in general, okay? There are some companies that don't do it, yeah? And then you have to do it by yourself. Okay, so I just wanted to mention just in case. Then yesterday you saw it with Guillermo. Then you get the FASTQ file. I think you are all already experts on this topic. Yeah, I think I don't need to repeat that much. But anyway, so FASTQ file, it's a 4 lines file where you have the sequence identifier, the nucleotide sequence, the quality score line that is separated by a plus, and then the quality score. Okay, so you have it here. So then you have this information. Then I wanted to expand a bit more on the Phred scores. The Phred scores are logarithmically related to the probability of an error. So here you have what Guillermo was explaining yesterday. So different Phred quality scores, okay? So Phred quality score of 10, 20, 30, and 40. So what that means, 10, 30, 10, 20, 30, and 40? Well, it means that if you have a quality score in your sequence of 10, you have one error in 10 base pairs, which is quite high. It's 10 bases in 100. It's a lot. So if you have a quality score in 100, it means one error in 100. Okay, so, and so on and so on. So if you go to quality score 30 or 40, the probability of having an error, it's going lower. Yeah, you got it all? Still all with me? Good. You already saw these plots yesterday, right? So I'm not gonna go much in detail. But here again, you have, yeah, well, one read, yeah, the x-axis is showing the whole length of the read, and here you have the quality score. And you can see that the quality of this read is extremely good, okay? So you don't have to do much here. Which software do you use for quality control? Well, yesterday we saw that Data2, the program, has already its own quality control. So there you are okay for 16S amplicon. But when you are dealing with a metagenome or with a genome, you need to have other tools. Here I listed some of them. The most widely used is FastQC. Okay, so I recommend. I mean, I only had experience with this one. I don't know, Luisa and Guillermo, if they have experience with the others, but yeah, this is the most widely used. Then again, I would like to emphasize a bit more here because this is an extremely important step before you start the assembly, okay? Because of course you get your reads, and what you want to get at the end of the day is your full genome or draft. Then when you are processing your data, yeah, you can see that sometimes you have, well, not so good quality. That's a signal of contamination, okay? Contamination with what? Well, it could be anything, but most of the times it's contamination from the adapters. The adapters that I show you from the demultiplexing, for example, are still there. Yeah? Or maybe primers or whatever. So then you basically need to run your tools to clean your data. And this is the sign. You will see that the Phred scores are going extremely down. So then now you need to take action there, correct the reads, and then rerun the FastQ program to determine if indeed you clean up your reads. So there are many tools. I just recommend here the ones that I have used in the past, and one of them, for example, is Trimomatic. Okay, I want to also say here that all the tools that we are going to talk today are not R-based. Most of them are Linux command line. Okay, so And we are not gonna see that in the practical today, not command line, because it's really time consuming, but Guillermo and Luisa, they will show you the process, okay? Okay, so we have gone already through the quality control and data processing. Is there any question? No questions. There is a question. The quality control and, sorry?
Quality score and
If there is any difference, okay, the question is if there is any difference between quality score and Phred score. No, actually, the Phred score is used to determine the quality. Okay, so what you see here in the y-axis is the Phred score. Okay, good. Okay, let's continue. So now we are gonna move to genome assembly. So we are gonna be increasing the complexity. So we are gonna see the first method of genome assembly or metagenome assembly, and then we will have a short break to clear our minds, and then we will come back, and then we will go through the other methods. Okay, so let's start. Okay, so I took this cartoon from internet, and well, you see that this was made in 2003 when the genome of the human genome was sequenced in 2001. And the problem that we had and the problem that we are having right now, it's indeed that we have too many pieces, too much DNA, too much information, and it's quite difficult and challenging to process all of that information. So you see this guy, this researcher here saying, okay, I think I found a corner piece. Most likely not, but it's quite complex. And one of the main problems that we are facing with all of these next generation sequencing technologies is that it's very difficult to optimize these technologies for highly repetitive sequence. And that's something that, well, we will have to deal with it because genomes are highly repetitive. Okay? So, and then what I'm going to show you is the basis for genome assembly taking into account this high repetition. The programs and the methods that I'm going to show you, they are not perfect, but it's the best that we have for now. Some definitions. Let's start by this. So we all know what is a read, right? Yes, thumbs up. Beautiful. So a read is a 500 to 900 long word that comes out of the sequencer. Then we have mate pairs. We all know what are mate pairs. Yes, we know, I think, because yesterday Guillermo showed. So a pair of reads of two ends that are coming from the same fragment. We also have contigs, yeah, because I'm going to be talking about these terms during the lecture, so I want you to understand exactly what I am meaning. So contig, it's a contiguous sequence formed by several overlapping reads with no gaps, okay? Then we have supercontig of scaffolds, so it's an order and oriented. That's very important, oriented, because, well, many times we don't know well the order of the contigs, yeah? And then, yeah, the oriented of set of contigs, usually by mate pairs. And then at the end, the final goal is to get the consensus sequence, okay? So how many of you had already experienced sequencing genomes or assembling genomes? Did you ever had any experience with genomes? Beautiful. 1, 2, 3, 3 only? 4. Good. It's good. Okay. Good. Let's continue. This is the whole process. This is the summary in a graphical overview. This is what is happening behind the scenes, behind the program, when you run the command line in the black screen. Okay, so first of all, you get the reads, and what the program does, it's going to do many iterations to try to find the overlapping reads. Okay, then next step is with these overlapping reads, you are gonna start forming contigs that we already defined them are longer sequence. Then with the contigs, you will make bigger contigs, so what we call scaffolds. Okay, so we will link the contigs and we will make these bigger DNA fragments. And then next step, and the best step, and where you feel much better, is when you get your genome. Hopefully a full genome and not a draft. Yes? Clear? Good. Let's start with the first method. Overlap, layout, consensus. Did you ever heard about this for the ones that already had experience? Yes, no? Yes, good. Well, the overlap layout consensus method works normally in 3 steps. It's basically what I already have described before, yeah? So it's first, it finds reads that are overlapping with each other. So it will iterate again one to each other, and then we will find the overlapping reads. The second step is where it carry out the layout. So it will lay out the reads and it will find complementary regions. Yes? And then the final step is when we are trying to build a consensus sequence. Okay? For now, this is pretty much what we saw before. It's just an iteration of reads that are matching to each other, right? And then at the end, we find the best matching sequence and the best and the longest sequence for us, okay, to finalize our genome. So this method, the overlap consensus, overlap layout consensus, was described by Staden in 1980, so quite recently. Maybe not for you, but for me. But yeah, so, and here is a paper where you can expand a bit more the information, okay? Then let's move on a bit into the repetitive sequence and how we could deal with it, okay? So here we have this example. So we have a region from a genome, okay? And then we have gene A, well, or region A, B, C, and then B that it repeated again. And then we have D. Yes? So then if we sequence, we get all of these different reads. Correct? But as you can see, these two regions are exactly the same. So if we are doing the assembly using this method, we are going to find a lot of problems, because how are we going to differentiate, yeah, if the reads are coming from here or from here or from here? It's going to be a bit difficult. So it will look something like this. So the first thing is that the assembler will get a bit crazy, and then it will not know what's the right order. So first, it will come through here. Then from here, we'll join these reeds to the red one. And then from the red, we'll go again to the yellow. And then finally to the blue. This is an easy example. Imagine when you have 10, 15, 20, 100 repetitions like that. Assemblers will not be able to resolve this. And then the result will be that you have, well, a lot of sequences that are not correct. It's everybody is understanding? Yes? Okay, so this is, for example, what happened with the human genome. Well, yeah, well, that was not funny at all because they sequenced the human genome and then, well, they found that most of the genome contains repetitive sequence. So it was extremely difficult to make the assembly. And yeah, even now, in 2022, we still get papers about filling the gaps in the human genome. So you can see how complex it can become the genome assembly. Okay, I don't know how are we with the time. We are fine. Are you okay? You want to have a break? No, we continue. Okay, let's move on to the next step. And I really want you to breathe and follow me. Please focus in the slide and in what I am saying because this is going to be a bit complex. It's very difficult to explain. I'm gonna do my best. Let's do it together. So let's move on to the string graphs or the brewing graphs. Josh, did I say well the brewing graphs? Brewing, yeah? Okay, yeah, more or less. So brewing, it's actually it's a surname. It means brown in Dutch. Okay. Well, let's move on to the genome assembly 2 string graph, the brewing graphs. So the first thing is that well, the brewing graphs works chopping the reads into shorter chimers. Okay. So this is how it works. So you have this read, and then it will divide or cut the reads, chopping the reads in shorter chimers. Okay, and then here you have like T, T, C, T, A, G, T. You are cutting them or chopping them in 3, and then they are overlapping to each other. Everyone got that? Good. So then now what it does, it builds a graph, what it's called the brewing graph. So every graph will contain what we call the nodes. And the nodes are containing the different chimers. So only the ones that are different, the unique ones. So then it will start building up your brewing graph, okay? And the connection between the different nodes, it's what we call the edges. Okay, so we are gonna go through an example and you will understand much better. So this is just the theory. So then finally, what we have to do when we build this kind of graph, we have to find the shortest Eulerian walk, okay? And then this is what it's called the walk graph. So we are walking through the brewing graph, and then we find the shortest path to get our genome. Okay, and sometimes, of course, you will have problems because you have repetitive sequence. No worries, we are going to do examples and you are going to understand much better. This is just the theory behind. Okay, so this assembler, the first assembler using the brewing graphs was published in 2001 by Pepner and Waterman. And again, more information you can find in this paper. So with this graph or with this method, rather than overlapping the reads, what we are doing is overlapping chimers. And I didn't define chimers yet, okay? Because I was waiting for next slide. So a chimer is what we define as a short and unique, that's extremely important, unique element of a DNA sequence of length K. And it's used by many algorithms, not only for algorithms for assembly, okay? So for example, here again, we have this example. We have a genome A, B, C, D, E, F, and H. And then what we do is we divide it in chimers of 3, and then we do the assembly. Here we have another example, okay? I want you to focus in this short genome A-G-T. And then we are breaking the rule of creating chimers here because I did it in purpose. So you can see if we create chimers of 2, yeah, we need to have AA, AG, GA, AG, and GT. So an AG, we are repeating twice. So that's wrong. But if I don't do it, yeah, I will not be able to resolve this because this is a repeated sequence, AG and AG, okay? So if we move on, and here you will see the power of the brewing graph to resolve these kind of problems. Because if we break the genome in chimers of 2, we cannot resolve it. But if we break the genomes in chimers of 3, then we can resolve it because then there is no repetition, okay, in the chimers of 3. So we will find the path to generate our genome. Yes? Thumbs up? Thumbs down? Yeah.
The timers of is an ideal length of element to be considered, or it may be more.
No. No, this is just an example. So I'm giving you an example, and Guillermo and Luisa, correct me if I am wrong. I don't think that any algorithm will do chimers of 3 or 2. This is just to explain. Normally, the chimers will be bigger because the reads are, 300, 500 base pairs. Guillermo is doing like this with his hands. The minimum chimer, it's 10, you said? Yeah, yeah. I will show you at the end, I will show you a script and then I will explain how assemblers work, okay? Good. Let's continue because I have more examples. I want to dig a bit more on this and I want you to understand the basis of this. This is what you do, yeah? Study and study and study, yeah? Right? This is what you do, yes. So, as you can see, it's highly repetitive, but we can identify the unique elements here. And I am already telling you what are the unique elements. So it's study and that's it. So imagine that we do sequencing on this. What do you get? Study and, right? Good. So can you try to assemble this using an overlap layout consensus? So the first method. What will be the result? Can anyone guess? Anyone? So remember, overlap layout consensus is what the wording is telling you. It's overlapping layout and finding the consensus. So what will be the result using the overlap layout consensus? This. So all the reads will get aligned like this, and there is no way to resolve it. Yes? All with me? Well, let's try to solve it using the brewing graph. So as I said, you need to find the unique elements from your genome or your sequence, okay? And then the two unique elements are study and that's it. So how to do it? Well, as I described before, you have to create what we call the nodes with the unique elements. And then now you connect the nodes with the edge, with the arrows, okay? So the first connection that we have to make is study and. Yes? The second connection is and study. And then again, study and study. Easy? Did you all get it? Question? As, yeah, so he's asking me, how do we know where to start? That's a very good question. I think what is happening is that the algorithm, it's running iteratively the same script or the same rule to find the best position to start. That's my guess. Okay, so it's like iteration. You know what is iteration? Repeating many times. Yeah, and we're gonna see another example now. Okay, any other question? Let's do the same with a small genome. So let's start with this genome, A, G, 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 A. Yeah? It's a short one, but for this example, it's gonna work. So let's build chimers of 3, trimers. So who can tell me which one will be the next chimer of 3? Oh, yeah, sorry, the effect. Now we have all of them there. Okay, yeah, that was right. AAG, and then the next one will be AGG, GGG, GGG, GGA. That's quite repetitive here, eh? So we are going to have problems there. So then let's try to build, yeah, the chimer graph, the brewing graph, but we are going to do it with chimers of 2 to make it a bit more complex and to have more repetitive sequencing there. So who can start? I only heard G, 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 G. Okay. I guess you are guessing, guys. Okay. Yeah? Oh, it's that. Yeah, I made a mistake. Okay. Yeah, they're making fun of me now. Okay. Yeah, that's a mistake indeed. Yeah, that's excellent that you spotted out. That's very good. You are understanding. Yeah. Yeah, it was on purpose. Yeah, yeah. Thank you, Luis. Okay, so let's build the graph now. So let's start AA. What's next? So the same. So it goes AA, AA, AG, GG, 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 GA. Is correct? Ah, that one is correct. Yeah. You got it more or less? This is a very nice question exam. I do it that with my students. Let's go the other way around. We get the graph and then we build the genome. AA, AG, GG, GG, and GA. You got it? Wow, I'm so glad. Thumbs up? That's thumbs down? Yeah? No? Okay, I think Kumar, this will be a nice question for the exam because they all got it. My experience is that 15%, 10 will answer well the question, but I'm sure that you will do much better. Okay, well, of course, I show you here a tiny example, but imagine having a full genome. Complex, eh? Well, this is how it looks at the Bruin graph of a genome. And I think it's a bacterial genome. So imagine in the human genome, or imagine a metagenome where you have thousands and thousands of microbes, thousands of genomes. So the complexity really increased. Okay? Well, and then, so one of my favorites, and I think Luisa is using in the afternoon, it's spades. There is even a version for metagenomes that it's called meta spades. And then just to give you an idea of the usage of this method for genome and metagenome assembly. So here I put the code for it to run spades, and the -k here are the chimers that are built. So basically what is happening is that every read is chopped into chimers of 21, then the 21 reads, 21 pairs reads, base reads, will start aligning to each other, and then we will move on to 33. We will chop the resultant assembly into 33, blah, 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 and so on, so on, so on, like that, okay? And then we will find at the end a consensus. Okay, so some tools to visualize these graphs. So when you do an assembly, most of the assemblers they give you the Bruin graph output, and you can open and visualize and play with it and try to find the best order of your sequence. And then you can use, well, here I put the link. The program is Bandage, and yeah, you can get these visualizations. Okay, good. There are many, yeah, you name it, assemblers. So here are some doing overlap layout consensus, and some others that are based on the Bruin graphs. And well, it's for me, I think we use in my group, we use a lot spades and meta spades for genomes and metagenomes, but also there is another one that is called unicycler, that it's for more for genomes, and you can use short reads and long reads together. There is another one called Fly that it's quite good as well, where you can use for mixed assemblies with short and long reads. Okay, good. Today in the afternoon, Luisa will talk about it. So once we get the genome, we need to do the quality assessment. So we need to assess how good is our genome. So for that, there are many tools as well. Which one is the best? I don't know. We use Quast. Okay, Quast is one of the tools that we use, and it will give you an idea, an overview of how complete is your metagenome or your genome, because this is used also for metagenomes. So it will give you the number of contigs, the total length, the M50 that today in the afternoon, Luisa will explain what it is, and many other parameters. Good. Guillermo? Maybe you can use the... One important is the length of your read, because if you read only, for example, 50 base pairs, you cannot choose a camera size of more than 50. You cannot. Yeah. But out there you can find like different formulas where you can estimate knowing the length of your read for the best camera size for your stuff. Thank you, Guillermo. Yeah, excellent. Indeed, doing a chimer of 100 in a read length of 50 doesn't make any sense. Good. Let's move on. I don't know, do you want to have a break? You want to continue? What do you want to do? Continue? Yeah, okay. So then let's move on to genome alignments now. So we will have a look at the 3 methods that are out there, and we will give some examples. Okay, so sequence alignment is the procedure of comparing 2 pairwise alignments or multiple sequence, and we can align genomes, we can align genes, we can align, whatever you have, okay? Here I show you a picture of how it looks like, for example, when you compare 2 genomes. So here you have the regions that are overlapping. So for example, purple here is overlapping a bit on a side. So here is genome 1, genome 2, and genome 3. And then you see also you can detect inversions, transverse, well, everything that is happening in the genome, you can detect it, okay? Here, for example, you can see an inversion. So this sequence flipped. So, and with, well, I will tell you which program was used for this, but this was WebAct. The alignment algorithms can be divided in 2. The first one is the global alignment, and the second one is the local alignment. Okay, global, it's also known as end-to-end alignment, and the goal is to align the sequence while maximizing the alignment score. So what that means? Well, it was described by Needleman Wunsch, and then what you do is you take the whole sequence, and then you use it completely, and then you just annotate if there is a mismatch or not. But you even take the gaps. Okay, that's the most important for this algorithm. You take into account the entire sequence for the alignment. The second method, sorry, there is a mistake here. So the second method is local alignment, and the goal of this one is to align the two sequences. So, and also that the alignment score is maximized. But as opposed of the global alignment, the final alignment will not contain the whole sequence. Okay, as you can see here, we took the whole sequence, even if there were mismatch or if there were regions that were not overlapping or even gaps. So, and no penalty is induced by the misalignments and so on. And then, well, here you see the example where we are discarding the extremes of our sequence. So we are reducing a bit the length, but yeah, it's containing still important information for the phylogenetic inference that is at the end what we want to achieve. So how does it look like in a scheme? Well, so the the local alignment will basically, when we compare two genomes, genome A in the y-axis and genome B, we can have that, well, the two sequences are completely the same or less similar or more similar to this other one. So, or there is an inversion or, well, a change in the order of the sequence. So this is what you get. So whenever there is a problem or a mismatch, yeah, this local aligner will take only the square ones, but will fill this with gaps. Okay, all of these parts will be filled with gaps. Positive about this, yeah, we can compare large genomic regions, so we can compare genomes using local alignments, and we can detect rearrangements like translocations or inversions and even sometimes duplications, but fails, for example, to detect insertions of sequence or even deletions. So how to interpret this other one is basically the same, but in this case, we are trying to detect the insertions and the deletions, yeah? And then we are, for example, here we have this sequence, so basically it put it here, and so on, this one here, and it will at the end try to improve the alignment. So we have that the global will detect insertion and deletions, but it fails to detect big inversions, okay? So then we come to the local alignment. So this is widely used in many of the programs that I will show you tomorrow for annotation. And it works like a mixture between the global and the local, okay? And then it will do something like this, taking into account as much as possible from the two alignments, okay? I don't want to go much in detail with this. I just want to show you that there are these three methods for alignments. Yeah, I think it's important that you know what is the theory behind and that you choose the correct one, or, well, you choose the tool that you want to use. So these are the tools that we use for whole genome alignment. So we have WebAct. I'm very fan of this one. It's easy, it's old, I know, but it's working very well. So it's from the Sanger Institute, and then you can see very well, for example, the red regions are completely aligned, the blue regions are moving around, or they were inversions or transversions or duplications, whatever. And then we have also Mauve. So Mauve, it's also quite old tool. And here you can even rearrange your contigs if the assembly didn't go well, and then you have regions that are not well aligned, and you know that they should be in another position. You can change and then save the genome in the right order. Then I wanted to give you a list also, although I think Guillermo will talk briefly about it. of alignment tools for also short genes or regions. All of these tools are command line, but some of them, they have the online version that you can use, although with some limitations. So I am really fan, for example, of MAFT. Where is MAFT? It's not in this list. It's not, no? No, no, well, no, yeah. It should be one of these. Yeah. Well, MAFT, for example, I look for the maximum number of positions that could handle, and it says that it could handle around 500,000 positions and a maximum of 500 sequence. So 500 length and 500,000 length, and then 500 sequence. I'm not sure, but this is what it said. And then, well, there are many others. I think you are all familiar with Clustal Double View or Muscle, or yeah, many others that are there. Some of them, as I said, they have they are available in the web servers. Okay, more or less. Yes, thumbs up, thumbs down. Good. Good. Yeah, good. Then let's move on a bit to the phylogeny, because now we have got our genome. We know the basis of the assembly. You are now experts in the Bruin graphs. So then you got your genome, and now you want to know what is this bacteria, and is there other bacteria that are similar? Are there other bacteria that are similar to this genome? Let's see how we can find out. Okay, so it's not the first time that you see this slide, and you will see again and again and again, because I think it's quite important that we always mention this amazing work. So, well, here to build this phylogenetic tree, they use 16S amplicon, thousands and thousands and thousands of 16S amplicon, 16S gene. But there are many other tools that can do a similar job with a full genome. Okay, so when I'm talking about full genome, I'm not saying that the whole genome will be included in the assembly. So that's what I call phylogeny all-in-one. So you have several programs. So like, for example, Amphora 2, Phyloflam 3, AutoMLST. So when there is a 2, it's because there was a version that was 1, and when there is a Phyloflam 3, there was a 1 and 2 before, okay? And there will be a 4, I guess. And of course, it has always improvements. So that's why you need always to look, and be updated with all of these tools. But just to give you an idea, the basis of all of these programs is, you all know housekeeping genes, right? Yes? Yes. So housekeeping genes are genes that are conserved among all bacteria, almost all. Some of them are missing, but one thing that is quite important is that these genes are not duplicated. Luisa will show you some details in the afternoon. And well, the difference between these tools is that they use different approaches to get this information. For example, Amphora 2, I remember the Amphora 1 was using 37 or 38 housekeeping genes. Now they increase it to 139 in the second version. So it's using 139 housekeeping genes that are in all bacteria, and they look for this algorithm look for those genes in your genome of interest. and then it will extract it, and then it will create the alignment, and then the phylogenetic analysis. So then phylophram 3 is the one that I like more. Okay, so this one I think is almost the only one that we use in our group, right, Guillermo? Also Amphora 2, I think. Okay, so but phylophram 3 is another all-in-one phylogenetic tool, and this one use I think more than 400 housekeeping genes. That doesn't mean that the 400 housekeeping genes will be included in your alignment, in your analysis. So it will search in your set of genomes. If you have 50 genomes plus your genome that you are interested in, it will search for the 400. And it will take the common ones and will give you a file containing those genes. It's quite easy to run them. And then you have AutoMLST. Correct me if I'm wrong, Guillermo. This one, I never use it. It's newer. But this one, you put your genome in the website. They have a web server, and then you will load it, and then you will see the phylogeny on the screen. You will see the result directly. Clear? Then we have other methods to estimate distance, phylogenetic distance. Yeah. So, for example, MASH. So MASH is a tool, and I recommend you to use it when you have many genomes. You can quickly run it, and then it will tell you how similar are all your bacteria based on the sequence that you are giving. Okay, that's another one, and we use a lot, and maybe Guillermo will also show a bit about it. Then visualization. Who is familiar with ITOL? Did you ever use it? Nobody? No? This is a very, very nice tool, and you can build, you don't need to have a server. You only need to have the phylogenetic tree. So basically the output of these tools. You load it, and then you can annotate your phylogenetic tree with the information that you have from your research. Imagine that you are working, for example, with endophytes and rhizospheric isolates, and then you want to see if all the endophytes are clustering together, or if all the rhizospheric isolates are clustering together, or if the rhizospheric and the endospheric isolates are the same. So you can use this annotation tool for that. And Guillermo this afternoon will show a small example and a big example, I think, yeah, where you can get lost. I'm sure that not, but you can play a bit with it. And the good thing of this program is that it has tutorials, and it has all the files online, and there is even a movie where it explains how to use it and how to upload, how to annotate, and how to do everything that you need to do. Another one, Graphland, from the Phyloflam researchers. So the Segata lab, he did Phyloflam, but also Graphland to generate these nice visualizations. This is a bit more complex, but I'm sure that you can also learn, because there is, there is documentation that you can read and follow the different steps. This is based on Python. Don't be afraid of Python. So it's all completely on command line. The main difference is that this one, although here you also need to create your files, yeah, this one is more intuitive. This one is command line. Okay, but I really encourage you to go next week when we are done with the course, and to explore, and to, if you have genomes already, to try these programs. Yeah. Okay. So I don't know how good are we with the time. Good. Perfect. So well, this is my last slide. So I wrote down this summary for you guys, and then you can see what we have learned today and what I would like you to keep from this lecture. So we have learned the first step when our meta genome, wherever you have, yeah, is sequenced. So now we know what we have to do. Yes? So we have learned what is the basis of the metagenome assembly, meta and genome assembly. And we know also some tools. And again, I want to emphasize that depending on your data, if you have PacBio, if you have Nanopore, if you have, well, long read, short read, you should use a different program. So read the tutorials for what is good, for what is bad, and then make your choice. We have learned about alignments, the different types of alignments, and some tools that you can use. Some of them command line, some of them online. You also choose. And then phylogeny and tools. And this afternoon, we are going to expand a bit more on the genome, metagenome assembly, yeah, the tools that we selected that we thought that it would be interesting for you based on your research, and some tools for phylogenetic analysis. Good. I think I want to stop here, and then tomorrow we will continue with annotation tools and comparative genomics. I think now it's a good moment to get ready for the practical this afternoon. So I think Guillermo and Luisa, they want you to download some files, right, Guillermo and Luisa? Oh yeah, sure. Yeah, sorry, I forgot. Questions?
Then I will talk a little bit about this afternoon. What will happen? Why don't you need something out of your green?
Maybe something, sorry, something to relate with your research, maybe, I don't know. If you have a genome, you don't know what to do with it. I love genomes. Give it to me.
Alignment using this feedback on movie. To that, the events of insertion, deletion, or translocation are recorded in that. But whether that is depends on the reference genome or it is the de novo one.
Well, what I was talking more was about de novo. Okay, if you have a reference, you are in an extremely luxurious situation because you have someone that already did it for you. Although when you compare genomes, you can see that even if they are close strains, there could be quite a lot of rearrangement. Yeah. So, but then the tools that I already show you, some of the visualization tools, you can use it to spot where the insertions or translocations or yeah. So you can look at MAUVE, for example. Do you have a genome that you are facing now that problem or?
I just got a reviewer comment that on what basis that inversion event has been included in the.
Yeah. Okay. Good question from the reviewer. I was not the reviewer, by the way.
maybe, okay, just the procedure, right? So what we're going to do is 10 past 12, so we get stop from the end of but today we also have a second step in the culturing of the microbes, right? And we're going to do activity profile. Muy claro.
¿Quieres usar esto? No, no, no. ¿Cómo ha quedado el...? ¿Lo tienes ahí? Muévelo.
So that
way easier. We're not gonna open R in any moment. So we're gonna show like still some commands and everything. So for today exercise, really simple what you have to download. Just download the folder assembly day 3. That's the only thing you need to download. So download the full folder that contains 2 other folders, the Power and 2 PowerPoints. Okay? Yes. So I repeat it. You go to the Google Drive that we share with you and just download in your computer the folder assembly day 3. Ya, pero para que primero vean aquí. ¿Mm? No, todavía no. Se me ha colado porque lo sube. Material that we will need for the practical of today. So it will be a bit more simple today. Okay?
Maybe one thing about the lecture material, lecture that we show in the morning. Maybe there are some files already in the shared drive. Don't download those. What we will do, because we are like cleaning them up, we will provide you with handouts of all the lectures at the end of the course. Right? So that will be then you have all the lectures in a handout format you can go back and go. Okay, shall we do it like that? All right.
Did anyone have any problem to download it? Sorry? Yes. Oh. So this is the main folder that we have. So you just need to download the folder that says assembly day 3. Yeah, the entire folder. Please let us know if anyone have any issue or any problem downloading it. Bueno, no te como desconectado de internet. Ya, perdona, se me han colado antes.
Yo, yo tengo un montón de notas que son mías. No sé, no sé, perdón.
Okay, did everyone manage to download it? Then we can go to the lunch break, right? So Kumar, please.
Okay. What's our plan now?
We can also take plates out of the incubator, bring it to the lab, we can organize there as well.
Yeah. The bioinformatics practical, the downloading part is still going on. So keep the data ready for today afternoon practical, right? So next thing we are going to move to the lab, take out the plates. So we complete that counting part before we break lunch. Okay. There is another small announcement. I think the nano drop reading, whatever you have taken the other day. Samples, I think, going the song and check. I think there's a lot of DNA there in the sample. Yeah. So there is. Yeah, perhaps you have to do the rechecking the sample. Yeah, you follow the instructions of Govinda Swami. Exactly. Yeah, it's a more of a kind of handling error. So yeah, exactly. I think most of the groups are still having the sample here. So you take the sample out and go for reading once again. So yeah, exactly. Sure. Yeah. Let's do that. I think that was already checked by Govinda Swami. So yeah. So there are 2 tasks right now. There are 10 groups, and go back to your incubator, take out the plates, start counting the colonies, make a record of the colony counts, whatever you are seeing on the plate. If you want, you can take photographs. All right. So do that exercise, and parallelly, you please take the count of your, take the quantitation of your DNA as well. Yeah. For online viewers, we are going to move to laboratory for continuing the practicals, both wet lab, that is the colony count, as well as the DNA quantitation. So join back at 2 o'clock for the bioinformatic practical.